start because I do have a few things I'd like to start by saying. First, let me begin by welcoming everyone. I think we'll get started just being mindful of everyone's schedules all over the world, wherever you're joining us from. My name is Jackie Kwan. I'm a neuroinflammation researcher here at the University of British Columbia in Vancouver, Canada. And I want to begin today by recognizing the International Women in MS organization really for their support of this session today. The network was, for many of you, you might be aware, it was developed after a first informal meeting at the American Academy of Neurology in 2018. And since then, it's grown to more than 300 members in over 30 countries all over the world. And their goal has really been to create bridges between junior and senior investigators, roads between peers, and avenues across various parts of the world. And it has been quite an interesting and I think rewarding initiative that many of us have been taking part in. In addition to a mentorship program and in-person networking events, iWiMS is pleased to support really a large number of scientific and clinical groups shown here, and those you can actually take a better peek at on the website. But today's session is the kickoff to an interest group that is perhaps among the broadest. You can see that we really have a bucket list here. And I'm very pleased to co-lead this with both Maria and Hedwig, both of whom are shown here. And really our hope is to invite host speakers that span both foundational and clinical aspects of MS research focused on many of these areas. And so for today's session, I understand some of you may need to leave early. We are recording today's session and it will be posted to the iWiMS website in the future. And in terms of format, we're asking our speakers to limit their presentation to 30 minutes so that we can allow for at least 10 minutes for questions both between and at the end. We ask that you please post questions to the chat and we will try to monitor it and address it in order as best as we can. If we don't have a lot of questions, it would be great to hear from the individual attendees themselves. So next up, we've got a terrific duo up today, and we're starting with Catherine Larochelle, a fellow Canadian who's familiar to many of you from her involvement both with ACTRIMS as well as elsewhere. Here she is up in the corner, shown enjoying the Rockies. Catherine is a clinician scientist in Montreal, and she's going to be speaking us today about her research on the deleterious crosstalk between oligos and T cells during neuroinflammation. Thank you so much for helping us to kick off the session. Why don't you go ahead and share slides, Catherine, and thank you again.
Thanks. Well, thank you so much for the introduction and thank you for the invitation. So I'm really honored to be speaking here today, and especially speaking before Bibiana Bielekova. It feels like opening for Beyonce, so I'm really happy to be here. So I will start, and I think that now it should work. Is it okay? Okay. Okay, so first I will just introduce my training as Jackie asked me. So I trained as an MD in Université Laval in Quebec City, and soon after joining the MD, I started a master degree in neurobiology. And at that time I was working on Parkinson's disease, and then I completed the MD MSc, went on with a neurology residency at Université de Montréal, and then I was missing research too much, so I did a PhD with Alex Prat on adhesion molecules expressed on T cells, and then went to Germany in Mainz to do a postdoc with Frauke Zipp on interactions between T cells and glial cells. And I am back in Université de Montréal since 2016, where now I have my own research group with excellent students, so I'm very fortunate, and a lot of help from Nathalie Arbour, Alexandre Prat, and Jack Antal, so I'm very fortunate to have lots of people supporting me. And this is the result of all of these interactions and colleagues. So I will talk to you about the direct interaction between T cells and oligodendrocytes in neuroinflammation. So here are my disclosures, so nothing related to the presentation today. So just a brief reminder, because I think everyone is very familiar with multiple sclerosis. So you know it's a chronic neuroinflammatory and neurodegenerative disease of the central nervous system. And you know that the diagnosis is based on the demonstration of dissemination in time and in space of demyelinating lesion. And I hope that you can see my pointer here. But here you have an example of an MRI showing typical white matter demyelinating lesions. And on MRI, we're much better imaging white matter. But in fact, there is also myelin in the cortex and in the gray matter. So you can see that you have lesions both in the gray and white matter in MS. And if you take a closer look, you will see the pathological hallmarks of MS, which is loss of the myelin signal. You can see here that you lose this blue signal, so it's sulfase blue staining, should stain myelin. And if you were also to take a closer look, you could see neuroaxonal injury and glial activation. And you see that these demyelinating lesions happen to form in the vicinity of small vessels showing perivascular immune cell infiltration. So here you will find T cell, B cells, myeloid cells. So because of that, we can say that myelin injury is at least partially immune mediated in MS. So of course, this kind of lesion in the early phase of MS, so in the very inflammatory early acute phase of MS, can lead to relapses and to clinical symptoms. So this is already bad. But what is also bad is that the myelin debris will impair repair processes. So myelin debris impair remyelination. So this means that not only can they cause symptoms, they can also inhibit the recovery. And what is also quite a problem is that, in the long term, if you don't have your myelin sheet around you as an axon, it can lead to neurodegeneration, because the oligodendrocytes also provide metabolic support that is very crucial. They decrease the energy demand on the axons, and they also contribute to learning processes. So this means that the myelin injury and the injury to oligodendrocyte could also contribute to the progressive phase of the disease, not only to the early phase. So it seems like it would be a good idea to try to protect them. And the good news is that the human mature oligodendrocytes are much more resistant to stress than rodent oligodendrocytes or immature oligodendrocytes. And in fact, it was shown by Dr. Entel, for example, here, and also by other groups in human and non-human primates, that if you stress oligodendrocytes, they are able to resist to some extent, and they will stop to extend their myelin processes. But if the environmental condition are improved, or if you restore favorable conditions, they will be able to re-extend their processes and try to help to remyelinate. So this means that it would be a good idea to try to protect the mature oligodendrocytes from the immune-mediated injury in MS. This could be a good strategy for neuroprotection. But then the question is, from which cells should we protect the oligodendrocyte, because it's still not clear exactly what are the sequence of events that lead to this myelin injury and oligodendrocyte injury. So if we look at the genetic risk factors that are associated with MS, the biggest risk factor is associated with the MHC class II. So this is implicated in antigen presentation to CD4 T cells. And if we look at the animal model of MS, this is also a CD4 T cell driven disease. So this is the active EE model, where we immunize mice with myelin antigen. And if we either do it actively, or if we then harvest cells and reactivate the cells, we can transfer the disease. However, not all the T cells are equal in MS or EAE. So if we skew cells towards a Th2 phenotype using AL4, it has been shown by many groups, but lots from Frauke Zipp, that in fact AL4 can be kind of protective and even promote neuronal regeneration or extension of processes. So Th2 are generally considered beneficial in MS, while Th17 cells, especially when developed in the presence of AL23, are considered pathogenic or very pro-inflammatory in MS, and they can induce damage and interact, for example, with neurons. So the question was, if we use a model that is not T-cell driven, for example, Cuprizone, well, and this was not done by us, this was previously published by the group of Dr. Calabresi. So you can see that even in the Cuprizone model that is not a T-cell driven model, you don't need T-cells in the Cuprizone model because it's a toxic injury to myelin. You can see that if you transfer Th17 cell, you will lead to worse demyelination, or you will impair remyelination. And in the end, Th17 cells seems to make matters worse for myelin and oligodendrocytes. So based on this, we thought that it would be really interesting to investigate if Th17 cells can really directly interact with oligodendrocytes and cause damage, or if it's just an indirect action so that they are recruiting other immune cells and then this leads to injury. So the first question was to say, can inflammatory CD4 T lymphocytes directly interact with oligodendrocytes or myelin processes in neuroinflammation? And we asked the question because most oligodendrocytes do not express MHC class II, so this means that this precludes the classical interaction with CD4 T cells. So there are some reports saying that 0.5% or 1% of oligodendrocytes could express MHC class II in inflammatory conditions, but this is very low. And therefore, we were looking for other ways for proving that there would be interactions indeed. So to answer that question, we first asked, do Th17 cells in vivo directly interact with oligodendrocytes? So to prove that, or to answer that question, we use a transfer model. So we transferred cells that are labeled with fluorescence, so either blue or green cells, and we transferred cells that were myelin specific or cells that were just polyclonally activated and would enter the CNS because the BBB was already destabilized by the myelin specific cells. And we transferred these cells in recipients where we had red oligodendrocytes, so either because they are transgenic mice expressing RFP protein driven by CNP, or because they are mice where we add Nile red, so this will stain the myelin sheath in red. Now, this will induce EAE, and when the mice start to show symptoms, we would proceed with live imaging. So the mouse is under anesthesia, and we proceed with 2-photon intravital imaging of the brainstem. So you don't have to break vertebrae or anything. You just do a hyperextension, you remove the meninges, and you can really image the brainstem of the mice. So this looks like this when we image. So you have in red the oligodendrocytes, so you see the cell bodies, you see the kind of sausages that would be myelin sheets, and in green here you see myelin-specific Th17 cells, but the behavior is relatively similar when you have polyclonally activated Th17 cells. This is a movie, you can see the time here, so this is a movie of 70-something minutes, so it's much accelerated, but you can see, for example, this T cells is interacting very intensely with one part of an oligo, then the other one goes back to this oligodendrocytes. If you look here, you have a Th17 cell that is interacting with the same part of oligodendrocyte for the whole movie. You have other ones that are kind of crawling or scanning along the myelin sheet, and some that find their oligo just at the end of the movie. So we decided to quantify these interactions, and what we found is that about 30% of the contact lasts for more than 5 minutes and 20% of the contacts for more than 10 minutes. And in fact, half of the cells of the Th17 cells that we were able to image would make such prolonged contact of more than 10 minutes. So this means that, yes, the Th17 cells can form prolonged stable contact with oligodendrocytes in vivo. And now the question is, does this mean something? Does it do something? So yes, it does, because if we compare the mean track velocity or the RS coefficient between Th17 cells that have made contact or no contact or were just scanning, we can see that there's a difference in the velocity and the RS coefficient. So it means that these interactions are biologically significant. And it is not based on antigen specificity, because when we compare the MOG-specific Th17 cells with the polyclonal Th17 cells, we can see the same type of contact. So we have the same percentage or similar percentage of polyclonal or antigen-specific cells making contact with oligos. And this was also demonstrated with using OVA-specific T cells. So now this means that in vivo, in EAE, Th17 cells are able to form significant contact with the oligodendrocytes, and we wanted to see if it could be relevant for MS. So we looked at MS tissue, and you can see here that both in the normal appearing white matter and in MS lesion, we do find CD4 T cells that are in direct contact with oligodendrocytes. So you have in green the CD4 and in red the oligodendrocyte, and I was very surprised at how easy it is to find T cells that are completely oligos. And here you can see, for example, this T cell that seems to be kissing the oligo. So about 10 to 20% of the CD4 T cells that we observed in MS tissue were in close contact with oligodendrocytes. So it seems that it is relevant for MS. But now we really wanted to be able to image something in a completely human setup. And of course, we cannot have a human under a 2-photon microscope. So my brilliant PhD student, Hélène Jaman, came up with a completely human setup where she used oligodendrocytes in primary culture that are derived from the resection path for neurosurgery for epilepsy. And this is in collaboration with Dr. Jack Antel and Chaolin Cui. So they isolate these oligodendrocytes, then we leave them for a couple of days so that they can extend again their processes. And then Hélène stains with a live dye so that she can see the oligodendrocytes. And in parallel, she culture human T cells. So she activates memory CD4 T cells with anti-CD3 and anti-CD28, and she adds either IL-23 or IL-4. So we call them Th17 and Th2, but it's really like IL-23 polarized and IL-4 polarized activated T cells. And then we co-culture these cells with the oligodendrocytes, and we do live imaging. So this looks like this, where you have the human T cells in green and the human oligodendrocytes in magenta. And if we follow the track of these cells over 30 minutes, what you can see is the Th2 cells seems to go further along, they go faster, while the Th17 cells, when they find an oligodendrocyte, they stay there and they interact quite intensely. Now, if we quantify that, as you can see, we see that the Th17 polarized cells form longer contact duration, are more likely to form long contact with the oligodendrocytes. And if you look just at the contact that lasts for more than 10 minutes, the Th17 cells do more static contact, which is like really staying in contact with the same part of the oligodendrocyte for the whole 10 minutes. And when we look at the velocity of these cells, you can see that mostly Th17 cells are slower than Th2 cells, even when we look at the contact that are more than 5 or 10 minutes, and also in static behavior. And this means that the interaction are more intense for Th17 cells than Th2 cells in this human setup. Now that we've seen that, yes, it does happen in vivo and it seems to be relevant for MS and in humans, we ask what are the consequences of these interaction between the Th17 cells and the oligodendrocytes. So to answer that question, what we did is to use again our human oligodendrocyte in primary culture. So you can see that they extend nice processes when you leave them for 6 to 10 days in their well. And then we decided to add Th17 polarized cells. And what you can see is that very quickly you lose the processes. You see the cells are still there, but you don't have any more than these nice processes. And this starts within 6 to 12 hours of co-culture. While when you put Th2 polarized cells, yes, you do have some loss of processes, but you still have lots of oligodendrocytes still showing nice processes. And when we quantify this, we can see that we have indeed much more damage when we put the Th17 cells than when we put the Th2 cells. Now, we were wondering, is it something that the Th17 cells just secrete, or is it really because they are in contact or in closer contact with the oligodendrocytes? So to try to understand if there is a difference between just being there or being directly interacting with the oligo, we came up with a setup where we use these same human oligodendrocytes in primary culture alone, or in direct contact with the T cells, or with T cells, but separated by an insert with a very small pore so that the cells cannot migrate, but anything they secrete can go and reach the oligodendrocytes. So using this setup, what we found is that the worst condition is really direct contact with Th17 cells. That is what induce the highest percentage of oligodendrocyte cell death. And this cell death starts around 12 to 24 hours after the co-culture. Now, we were wondering, this is nice when we do it like in a kind of artificial way, comparing Th2 polarized to Th17 polarized cells, but then what happens if we compare T cells from MS versus controls? So to do these experiments, we use all CD4 T cells isolated from MS or controls, and we just activated with anti-CD3 and anti-CD28, so no cytokine to skew, just like activated T cells. And when we co-culture these T cells with our human oligodendrocytes, what we see is that there is damage with both the healthy control and the MS, but this is significantly worse when you co-culture with T cells from MS, because the length of processes is significantly shorter after co-culture with MS T cells. So then this means that indeed the T cells, and especially the T cells from MS, can harm oligodendrocytes. And of course, then the next question was, how do they induce this myelin injury? So based on previous studies, we know that if you delete one of the mTOR receptor subunit, this will change the calcium influx in the oligodendrocytes, and this will, in fact, kind of protect the oligodendrocytes and the myelin sheath. So if you delete this mTOR receptor subunit in oligos, you have a better preservation of myelin sheath and a better preservation of axons in EAE. So it seems like glutamate could be one of the mediator. And what's the link between glutamate and Th17 cells? Well, the group of Frauke-Zipp and Stefan Bittner had just shown in 2019 that Th17 cells have all the machinery to release vesicles of glutamate. And in fact, they had also shown that Th17 cells from MS patients release more glutamate than Th17 cells from healthy control and more than from Th1. And they had also identified that beta-1 integrin or CD29 interaction with the ligand will trigger this release of glutamate from vesicles in Th17 cells. So then we thought, could glutamate be one of the toxic mediator? So first thing we did was to put glutamate on our human oligodendrocyte in primary culture. And what you can see here is that indeed, if you put glutamate, you will lose these processes from the oligodendrocyte, which is not the case when you put, for example, inflammatory cytokine TNF and interferon gamma. And Chaoling Cui, who works with Dr. Entel, has performed this very nice ensheathing assay where the human oligodendrocytes can ensheath nanofibers in wells. And you can see that glutamate does impair this ensheathing capacity. Now we said, then glutamate is toxic, but does it really come from Th17 cells in our setup? So first things first, we just checked that indeed the Th17 cells are better equipped than Th2 to release glutamate. So here you have VGLUT1, which is a vesicular glutamate transporter, and you see that it's much more expressed in our Al23 skewed than our Al4 skewed T cells. And you can see also that our Th17 cells express higher levels of CD29 or beta-1 integrin that is implicated in this vesicular release from Th17 cells. This is true in human and in mice. And then we were able to confirm that some of the ligands of CD29, so fibronectin, MADCAM, and COL4A, are expressed by oligodendrocytes. So it means that indeed everything is there so that they could interact together through CD29 expressed on Th17 cells and one of the ligand expressed on oligodendrocytes, and that this could trigger release of glutamate. So we were indeed able to measure glutamate in the culture medium, but most interestingly, we were able to show that if you block CD29 on T cells, you will protect the processes of oligodendrocytes that are in co-culture with the T cells. While if on the contrary, you use a CD29 agonist, so which should increase the release of glutamate, you will see an increased destruction of the processes from the oligodendrocytes. And this is just a quantification here. So globally, we were quite convinced that Th17 cells can release glutamate when in contact with oligodendrocyte, and this will lead to injury to their processes. However, even if we leave glutamate for 48 hours, we don't see cell death. We just see this kind of retraction or a non-maintenance of the processes of the oligodendrocytes. So our next question was, how do the human Th17 cells induce the oligodendrocyte cell death within 24 hours? Because we do see, first we see injury to processes, but after that we do see that Th17 cells can kill the oligodendrocytes. So to try to have an idea of which pathways can be implicated, with the help of our excellent bioinformatician Olivier Testé, we perform and analyze single-cell RNA-seq of human Th17 cells and human oligodendrocytes in primary culture that were either in direct contact or separated by an insert, as I've shown you before. And as I've shown you before, in this condition, we have much more cell death than when separated by an insert. So what we got was this kind of UMAP where you can see clusters of cells. And if you look at the genes that are expressed, you can very easily see that on the left, you have the T cells that are expressing these CD3 genes. And on the right, you have the oligodendrocytes in green that are expressing PLP, CNP, MOG, etc. So if we first look at what happens to oligodendrocytes when they are in direct contact versus separated by an insert from Th17 cells, now we reclustered focusing only on oligodendrocytes. And what you can see here is that cluster 1, that is in green, is much more abundant in the oligodendrocytes that were in direct contact than the ones that were separated by the insert. Now, if you look at the genes that are expressed mostly in the oligos that were in direct contact versus the one that were separated by an insert, you can see that contact with T cells will increase the expression of chemokines CXCL10, CXCL11, and CXCL9, and also of SOD2 that is implicated in the process of reactive oxygen species, while on the insert side, you have, for example, NKX62, which is implicated also in myelination. And if you go with the geo pathways that are implicated, so in green, you would have what the pathways that are kind of enriched after contact, and in orange, the one that are greater after you got separated with the insert. So you can see that if you leave the cells not in direct contact, so separated by the insert, you have more ensheathment of neurons, neuron differentiation, glial cell differentiation. So more like towards the idea of continuing to try to myelinate or maintain your processes. While if you are in direct contact with Th17 cells, the oligos will upregulate cytokine response, innate immune response, and you see even regulation of cell death and apoptotic process. So this was on the oligo side. Now, if we look at the T cell side, and we focus on the cells expressing the CD3 genes, again, we recluster only based on T cells. And what you can see here is that you do have big differences between the relative abundance of cluster when you compare Th17 cells that have been in direct contact with oligo versus separated by an insert. And in particular, in direct contact, you have a greater proportion of these cluster 0 and 2. And if you look here at the top genes that are expressed in these clusters, you see that cluster 0 is associated with Al17 interferon expression, and this cluster 2 is associated with perforin and granzyme expression. So it looks like inflammatory and cytotoxic T cells. So kind of the genes that are associated with these subset are enriched after direct contact with oligodendrocytes. Now, if we look in more details, we decided to see, do we have a common core? So among all of these 4 clusters that you can see here that were identified in the T cells, if we compare contact versus insert, what are the genes that are all upregulated or downregulated in all of the clusters after contact? And we came up with 71 genes. And if we look at the processes that are related, we see that it's interferon gamma or type 1 interferon signaling. We see there are some metabolic processes, regulation of regulatory T cells. So quite interesting from an immune point of view. And when we looked at the top genes that were upregulated after direct contact, one of the genes that was the most interesting was granzyme B. So this was upregulated in all T cell clusters after direct contact with oligodendrocytes. And we could confirm using ELISA on supernatants of the co-culture that indeed, after contact with Th17 cells, there is much more granzyme B in the medium than when separated by an insert or when you compare with Th2 cells. So as everyone knows, granzyme B can be cytotoxic, so can be used to kill cells by T cells. Usually it's more associated with CD8 T cells, but it is also associated with CD4 T cells. And here what we could show is that if you image these co-culture of T cells with oligodendrocytes, and you have in green the Th17 cells or the Th2 cells, you have in magenta the oligodendrocyte. You see one that is really not feeling well and is dying here with normal oligos around. And you see in yellow the granzyme B. So you can see that you have granzyme B in the Th17 cells that is polarized toward the oligodendrocyte that is now containing granzyme B also, and is the oligodendrocyte that is dying. Well, if you look at these Th2 cells, you don't have that much granzyme B, and it doesn't seem to be polarized towards the oligos. So finally, to prove that granzyme B is indeed implicated in the Th17-mediated cell death of oligodendrocytes, we use MO313 cells, that is a human oligodendrocytic cell line, because the human oligos are not very happy when you put the MSO. We use this cell line, and we use a blocker for granzyme B. What we were able to show is that if you use this blocker, you will protect the cells from the Th17-mediated cell death. So globally, what I've shown you today is that we were able to show first in vivo that there are direct contacts between Th17 cells and oligodendrocytes, and this is not dependent on the antigen specificity as would be expected, because we don't really have lots of MHC class II expressed on oligodendrocytes. Then I've shown you also that we can see such contact between T cells and oligodendrocytes in MS tissue and also in vitro in a human setup. And what we think might be happening is that when you have an inflammatory environment, it seems that oligodendrocyte would upregulate chemokines such as CXCL10. The T cells that have the receptor for this could be attracted to the oligodendrocytes. There are probably some CAMs that are also implicated, and that's what we're studying right now. And then when the Th17 cells comes near to the oligo that are expressing some of the ligands for CD29, we could see some CD29 triggered release of glutamate that would cause processes injury to oligodendrocytes, so we lose these kind of long processes, and then and this happens in 6 to 12 hours, and then after that, when the T cells have been in contact with the oligos themselves, they will change their profile, they will secrete more interferon gamma and granzyme B and IL-17, and then with this release of granzyme B, they seem to be killing the oligodendrocyte, which would lead in a second step to this oligodendrocyte cell death. So right now we're trying to identify other cell adhesion molecules that might be implicated in this deleterious crosstalk, and we think that it's a very interesting avenue to explore for neuroprotection. So with that, I'd like to thank everyone that has been implicated. So it's, of course, a lot of work, and especially Hélène Jaman has been leading this project, and Harita Desu, who's now my postdoctoral fellow, is also pursuing this line of inquiry. And we had lots of help from Dr. Entel, Alex Pra, Nathalie Arbault, Franck Zip, Stéphane Bittner, and our clinical team at the CHUM. So thank you for your attention.
Fantastic, Catherine. That was lovely. I would like to just see if anyone can just maybe put up their hand or just simply unmute. We're a small group to go ahead and ask questions. I think I've got one from Hedrick.
Yes, I can't see myself, but I think you can see me. Can you hear me too?
Yes.
Yes.
Okay. That was amazing work. I tried. Oh, my son is saying hi too. My son liked it a lot too. He was repeating TH1, so I was very proud. So this is beautiful. And I was wondering this. I mean, this is kind of a nasty question, right? But how would things change when you put other cells in the mix? So all these factors are released. So do you think all of this is still going on in vivo, or is some of it maybe buffered away by microglia or astrocytes or neurons? Any thoughts on that?
Yeah, that's a really good question. It's kind of difficult in the human setup to try to reproduce this. In mice, what was really interesting is because of the percentage of contact that we had with oligodendrocyte and the RS coefficient and everything, we can say that these T cells really wanted to interact with oligodendrocytes, and it was not only because because we were not imaging the microglia, so we were not seeing that they were interacting with them. So we think that it does happen, these contacts in vivo, because we see it. It's difficult to say if if all the glutamate or granzyme B could be buffered by astrocytes, but what we think it happens really in the microenvironment when the T cells are really in close contact with the oligos. So we think that it would be hard to buffer in such a small microenvironment. So that's what we think happens. So that's why we think that if we were able to stop this very close interaction, we could probably stop the cytotoxicity or the toxicity to the oligos. But for sure, in the real life of an oligodendrocyte, there is lots of interaction and other things happening, not just T cells.
Really cool. Thanks.
Thanks.
I see we've got another one from Bibi in the chat. Similar to what I said, Bibi is going to be asking, how about Th1 CD4 positive T cells? I'm going to ask about Th0 because they produce a ton of glutamate too, just on being activated. But anyway, back to Bibi. How about Th1 cells?
Excellent question. So yeah, if we had like an unlimited supply of oligodendrocyte, we would test all of these subsets, but we have to choose at some point because we don't have many. But you're absolutely right. So Th1 cells, Hauke has shown that they express lower amounts of glutamate. They do express some, but lower amounts than Th17 cells. So we think that if for glutamate wise, it should be less harmful than the Th17 cells. And also there has been some work, not by our group, but others, showing that I think Th1 cells are not interacting as much or not as toxic in vitro to some kind of oligo lines than Th17 cells. But that's absolutely something that would be interesting. And I'm not sure, in humans, I'm not sure that you can really say it's just Th1 or just Th17. I think that most of these cells are plastic, and at some point they might be Th1, and later on they look more like Th17. And as I'm a big fan of MCAM, and they are double positive cells, I think that the most dangerous cells in MS are the ones that are able to switch to interferon Al17, GM-CSF, and all of this. And in our case, also with this contact with the oligos, the cells also upregulate interferon gamma really highly. They do secrete lots of interferon gamma after seeing oligos. And we know that Th17 cells tend to switch to a Th1 phenotype after entering the CNS. So that might be one of the mechanism that would underline this switch. And yeah, so I think that Th1 cells probably are also deleterious, but maybe not as much as Th17 cells. So I see,
one more in the chat. Do you see CD4? This is from Cynthia. Cynthia is asking, do you see CD4 positive T cells close to oligos and releasing granzyme B and glutamate in active MS lesions? I remember some data for CD8 positive T cells only.
Yeah, that's also a really great question. It's not easy to do the triple staining for granzyme B, CD4 T cell, oligodendrocytes, and the nuclei in MS brain. So we do see some. We were able to identify some. I didn't put the image here, but we have images in the article. We do see CD4 T cells expressing granzyme B in the CNS of MS patients. We were not able to see that there was like really granzyme B in the oligodendrocyte. It looks like there might be some granzyme B in the oligodendrocyte that is next to it, but it's, as I said, it's not an easy thing to do. And for glutamate, I'm not aware that we were not able to really stain it in tissue so far. So I would have to, for the glutamate, I would have to say that technically, I think it was too challenging for now.
Catherine, before we move on, I have one quick question. For your glutamate co-incubation assays, or even with your T-cell co-incubation assays, did you ever come in with any different glutamate receptor antagonists and try to pinpoint what it was and determine that it was indeed specific to glutamate directly?
Well, we did try some NMDA receptor antagonist, I think, and I think it's more AMPA than NMDA. But again, this is challenging with human primary oligodendrocyte because there's lots of buffer or stuff that they don't like. So that even the control is a bit difficult. But we think it's probably more AMPA than NMDA.
Okay, all right. That makes sense, I guess. But okay. Do we have any other questions from anyone? You can just unmute and go ahead and ask. No. I don't see anything. So with that, why don't we move on to our next speaker? Thank you again, Catherine.
Thanks.
Okay, so next up is a researcher and clinician who is dear, but unfortunately not so near to me anymore. Someone who in many ways took me under her wing when I first arrived at the NIH several years ago. So it's my pleasure to introduce Bibi Bielekova, who's going to share with us her recent work investigating the biological basis of MS progression and severity. Thanks so much, Bibi. I think you can go ahead and share. Perfect.
Good. Can you see it?
Yes. Terrific.
Okay.
You're good to go.
Wonderful. Thank you very much for inviting me, Jackie. This is real fun. You know, I posted my email. If you have any questions or, you know, anything I can help you with afterwards, by all means, feel free to reach out. So Jackie said we have to say something about our training. So I'm actually from a small country called Slovakia, in the middle of Europe. And my elementary school was focusing on downhill skiing, which really taught me hard work and persistence. Then I went to high school, which was specialized on mathematics and computer sciences. So the class that was from whole Slovakia had 28 kids. And we had 24 hours of math and related disciplines every week. That cured me of wanting to do pure math for the rest of my life, but it did dramatically influence how I am thinking, and I'm very, very grateful for that training. Then I went to med school because we don't have college. I mean, we don't have college if you go to med school. We have 6 years of med school. And that was really the low point of my education in Slovakia. All I had to do was sit on my butt and memorize. But fortunately, after my 5th year, I went to SUNY Downstate for externship. And there I realized that medicine can be beautiful and creative and logical. And I totally fell in love with that type of medicine and decided to come to United States. So in my 6th year of medical school, I passed these predecessors of USMLE at the US Embassy in Vienna. And then I was immediately recruited to SUNY Downstate to do my internship. Then I did my neurology residency at Boston University. And then came to NIH as a neuroimmunology fellow, was there for 3 years. Then I was promoted to staff clinician, which is kind of glorified fellow. It's really the same thing. And then I realized that if I want to have my independent group, I have to leave NIH. So I did that, went to University of Cincinnati, was immediately associate professor with tenure. And pretty much as soon as I left, NIH started to call me back, and 2.5 years later, I went back, and I am there since. I moved from NINDS to NIID, which is now my home, and very nice home. So I'm gonna talk to you about biological basis of multiple sclerosis severity. All this was deposited to medRxiv and has been in review process and rejection process for a couple of months now. Of course, The work is a teamwork, and I really, for this particular study, I want to highlight Peter Kosa and some internal and external collaborators. So the multiple sclerosis field has been extremely successful in developing new drugs for MS, but as we have shown in this meta-analysis of MS clinical trials, their efficacy on inhibiting disability progression decreases as a function of age of the patients at drug initiation. And so after age about 53 years, this model predicts 0% efficacy on a group level. And that really leaves 45% of people living with MS today without effective treatments. Furthermore, I will show you that for additional 38% of patients, current treatments really provide marginal efficacy. And I will show it by demonstrating or showing you the raw data from this ocrelizumab clinical trial, which recruited people with a mean age of about 44 years, in fact, excluded everybody over age 55, and reported about 25% efficacy on inhibiting disability progression. And I'm focusing on ocrelizumab because it's one of our most efficacious, if not the most efficacious drugs that we have. What you can see from this oratorio clinical trial that ocrelizumab and placebo arms already achieve maximum separation at 12 weeks of treatment, which really tells me that the drug must be working on contrast-enhancing lesions, because that is the biology that can be inhibited so fast. And indeed, as you can see, quite unusually for progressive MS, a quarter of these patients had contrast-enhancing lesions. So I mentioned that at the time when the experiment was stopped, they reported 25% efficacy. So what does it mean? Does it mean that 25% of patients will not progress? Or does it mean that on average, every patient will progress 25% less? And it really doesn't mean any of these things. It's a relative number relative to when the experiment was stopped. Now, we can look at these data differently and derive absolute efficacy from these data by selecting any level of disability threshold and simply asking, what is the time delay that the ocrelizumab arm compared to placebo reaches this disability threshold? And as long as these arms are parallel, which they are, the absolute efficacy is 6 months, and it's 6 months for basically duration of the treatment of any individual patient. And really, that means that in middle-aged MS patients with substantial MS lesional activity, meaning contrast-enhancing lesions, ocrelizumab delays sustained disability progression by 6 months. Of course, what doesn't remain constant but becomes cumulative are side effects. And so I showed it to you because I want us to understand that therapeutic need remains really high for most MS patients, and that this need must target non-lesional MS activity. So what do I mean by non-lesional activity? Well, we really know what are causal processes of MS lesion, and furthermore, we know how to inhibit MS lesions. In fact, ocrelizumab does it by 95%. What is less appreciated that this lesional activity decreases simply by MS duration, and these data are beautifully documented in many different natural history cohorts. So even in the untreated stage, as people progress along this axis of time, they form less and less focal lesions. And that means that all of the disability progression that I showed you in the ocrelizumab arm is really due to non-lesional MS activity. People also call it PIRA, progression independent of relapse activity. And of course, pathology studies identified many different processes that happen in the CNS tissue, but the bottom line is we do not know which of these are causal. And that is what my lab is interested in. And our hypotheses are that the pathogenic mechanisms that underlie MS non-lesional activity are multiple, individually diverse, and likely evolve with disease progression. And if fully evolved, MS can be successfully treated, then it will require rational combination treatments that target most or ideally all disease mechanisms that are expressed in individual patients. And both of these hypotheses come from our collective experience with other human polygenic diseases, such as, for example, cardiovascular diseases. As you can hear, my dog, Beash, is really protecting me from all neighborhood squirrels, if you were interested. So, for cardiovascular diseases, we have more than 100 different treatments today. And of course, clinician assigns these treatments based on the measurements of these processes, which we used to call risk factors. But in fact, today we understand that they are truly causal processes and that they cause the disease through the dose exposure interaction. And we got this understanding by ability to measure these processes longitudinally in living people with cardiovascular diseases. And so I believe that analogously, if we want to understand which of these candidate processes are causal in MS, we need to be able to measure them longitudinally in living people with MS. And furthermore, we need to measure them in a way that assures that these measurements originates from central nervous system tissue, because you can see that many of these processes are in fact happening in any kind of organ. And really, I don't know any other way to achieve this but by studying cerebrospinal fluid biomarkers, and that's what we are doing in our natural history program. And then, of course, we don't have hundreds of drugs for this non-lesional activity. In fact, I believe we don't have any. And so we are running clinical trial. This is platform clinical trial, TRAP-MS, where we are validating the targets that come from our natural history protocol and also developing CSF biomarkers as companion diagnostics. So the design of the study that I'm going to show you today is really following the design of most of our studies. So the natural history protocol has prospective acquisition of subjects in an untreated stage. Everybody undergoes a thorough neurological examination. We collect 8 disability scales, 4 MS severity scales. We collect volumetric imaging of brain and upper cervical spinal cord, and we collect biological samples. For the CSF samples, we of course run traditional laboratory markers. We used to do multicolor flow cytometry from CSF. We now completely switch to CiteSeq and PenSeq that allow us to also get transcriptomic signature. And then we cryopreserve CSF cell-free supernatant. And everybody undergoes longitudinal follow-up. So by the time we were running this particular experiment, we included all CSF samples from untreated MS patients and healthy donors that were available in our database, which were over 1,000 different samples. These were analyzed blindly by DNA upcomer assays. So these are DNA molecules that are modified by side chains, which allow them to bind with high affinity and slow off-rate specific proteins. And this particular assay was able to measure close to 5,000 different proteins. And then we unblinded diagnostic categories. We used embedded healthy volunteer data to regress out effects of physiological aging and sexual dimorphism. And then we used bioinformatic approaches to identify pathways and upstream regulators that correlate with these different progression and severity outcomes. I will only be able to show you the pathway analysis. And finally, we assembled CSF biomarkers into machine learning models that predict 9 different outcomes. And of course, we validated these models in the independent validation. So very often we get questioned, what is the difference between using subtracting effects of physiological aging and gender dimorphism? Why don't you use MS age and gender as a covariate in the model? And so I'm gonna just showcase the difference on example of one of the proteins. This is modelin. And you can see that that protein in MS cohort significantly increases with MS disability measured by the Scombi-Wise disability scale. But this modeling also correlates with age in MS patients. And so if you just use as a age as a covariate, you will conclude that this modeling does not correlate with MS disability, that everything was due to the age. But of course, age also correlates with disability in MS. So instead, if you use healthy volunteer data, you see that this modeling still correlates with healthy volunteers, but the slope is much lower. So if you simply subtract the variance that is due to physiological aging, you will conclude this modeling still correlates with MS disability. I won't have time to introduce scales that we have developed over last decade. I'm just going to say that due to very small effect sizes that any single gene or any single protein exerts on a very complex outcomes, such as clinical outcomes of disability or severity, any success of modeling really depends on the outcome quality. And unfortunately, the outcome that are currently broadly used in MS field do not have sufficient sensitivity, linearity, or signal-to-noise ratio. And so we had to develop novel outcomes, and I'm just gonna say that what we like to do, we like to measure the same biology or the same process using different measurements. So here is outcome that we call brain damage, and you can see these are individual patients that are progressing differentially in this brain damage outcome. And what the brain damage outcome is, it's the inverted first principle component of brain parenchymal fraction. So this is a brain atrophy from the MRI images, and the symbol digit modalities test, which is a cognitive scale. So you can see that they correlate with each other. And when you take the first principle component, you invert it, you get this brain damage, which still correlates very highly with the outcome that everybody else uses, which is brain parenchymal fraction, but it has much better sensitivity, linearity, and signal-to-noise ratio. Similarly, this outcome called Combi-Wise, combinatorial weight-adjusted disability scale. It's a machine learning derived outcome we published a few years ago. It's a linear combination of these 4 outcomes. And the most important thing is that it correlates highly with EDSS, which is of course ordinal scale from 0 to 10, whereas the Combi-Wise is now continuous linear scale from 0 to 100. But it's really important for us to have this strong correlation, because I will show you that basically we can have best of 2 worlds. We can use these outcomes for modeling, but then we can simply recalibrate the models to predict these outcomes that everybody else recognizes and uses. All right, so when we modeled CSF biomarkers against 4 MS progression outcomes, we were quite surprised to identify many pathways related to neuroinflammation. And the second biggest signature were pathways related to fibrosis. And I said that we were surprised because I alluded to the fact that the contrast enhancing lesions decrease with MS progression. And because field equates contrast-enhancing lesion with inflammation, generally people believe that the inflammation decreases with MS progression, and here we are basically showing the opposite. So it's really important for us to validate these data, and we were able to do that in very unique cohort of longitudinal CSF exams. These are untreated MS patients that basically participated in our previous placebo-controlled clinical trials, and they were in the placebo arms. So now, simply calculating yearly change in these CSF biomarkers and putting that into the bioinformatics pipeline, we were able to validate exactly the same pathways with very low p-values. Highlighting PPAR signaling because it's one of the two negatively correlated and validated pathways, because we are using pioglitazone PPAR agonist in the TRAP-MS protocol. And I'm also highlighting one of these fibrotic pathways and showing you, in fact, these longitudinal untreated MS patients and their CSF data showing how this pathway increases with MS progression. And this was rational for testing pirfenidone, which is anti-fibrotic drug in TRAP-MS protocol. Now, having these diverse MS progression outcomes, we thought that we can differentiate biology related to spinal cord injury from biology related to brain damage. And we thought that we knew that this brain damage outcome combi-wise, in fact, correlate with each other. Yet, if we take the residuals, we would say these people have proportionally higher physical disability, and these people have proportionally higher cognitive disability. So we can take these residuals and call it spinal cord disability. And indeed, this does correspond to what we see on the MRIs of individual patients. So this is patient 1, and you can see pretty good brain, but severely affected spinal cord, and vice versa, patient 2, brain atrophy and nice and juicy spinal cord. But is it true on a group level? So here is kind of the same data I'm showing you first and third quartile, and we use propensity score matching to take patients from these different quartiles and match them for the brain damage. And as you would expect, they were highly different in the spinal cord damage. And we can do the same kind of opposite and match for spinal cord damage and have vastly different brain damage. And then we did a volumetric analysis of the upper cervical spinal cord, and we demonstrated that indeed these residuals do correlate with the spinal cord volume, and that people in this upper quartile have significantly lower spinal cord volume than these people in the first quartile, as you would kind of expect. So then we simply asked what are the FBR adjusted significant differences in CSF biomarkers between these quartiles, and put that into the bioinformatics pipeline. And what we identified was that people with predominant spinal cord damage had activation of fibrotic pathways, whereas people with predominant brain damage had overall activation of neuroinflammatory pathways. And you can see that these are almost mutually exclusive. Okay, so I showed you biology that correlates with MS progression. But, you know, this is kind of, this type of analysis cannot differentiate epiphenomena, which are processes induced by MS, from processes that are truly causal. So to get us closer to the causal processes, we would love to be able to model biology against these individualized progression slopes. Practically, that's impossible, because the moment we diagnose MS, we put people on treatments, and different treatments affect these progression slopes differently. And so instead, we must use what everybody else is using, which are cross-sectional measures of MS severity, where we take patients of the same age or same disease duration, and we differentiate those that accumulated more from those that accumulated less disability. And if we do it across all of the different ages, then indeed we select patients that progressed faster from patients that progressed slower, just looking back in time when they were still untreated. And again, I won't show you all of these different outcomes that we developed for this analysis. I will just highlight MSDSS, it's published, because to my knowledge, it's the only scale that truly predicts future rates of disability progression in the independent validation cohort. So now when we modeled CSF biomarkers against these 4 MS severity outcomes, we again pulled out inflammation, but slightly different inflammation. So first of all, for the first time, we pulled out pathways related to B cells and plasma cells, which of course we know that's the hallmark of MS, and pathways related to IL-17. It was interesting to hear the previous speaker. We also pulled out 3 pathways related to viral infections, very interesting in view of EPV connection to MS. And from innate immunity, we really pulled out pathways that are focusing on effector mechanisms such as pyroptosis, natural killer cells, and production of nitric oxide and reactive oxygen species. We did not see any inflammation-independent neurodegeneration. In fact, all of the CNS pathways that we pulled out were negatively associated with MS severity, which suggests that they are neuroprotective. And indeed, you can see synaptogenesis, Narayan signaling are neuroprotective pathways. And finally, the neuroinflammation associated with
Hi, Bibi. Sorry, I'm going to interrupt for a sec. I'm missing the last part of what you just said. I don't know if others are having sound issues or not. Can maybe some people unwind and let me know if they're finding the sound a little bit spotty or not? Anyone?
I hear well.
Okay, perfect. Sorry.
It's disappearing a bit off. The end of the sentence has disappeared for me.
Yeah, okay, Helen, maybe it's a Vancouver thing. Okay, here we go. Line was not stable. Perfect. Okay, let's see how this goes moving forward. Bibi, if you don't mind popping back to the synaptogenesis signaling, I've seen that was certainly on your last slide, a big outcome to see that it wasn't, or that it was negatively impacted. I think that was the point that I missed, and maybe Helen as well.
You hear me better, Jackie?
Yeah, that is better. Does it work for you?
No, it's not gonna.
It's okay. You know what? I think we're okay. It was just towards the end of the sentence there. Let's maybe, if you don't mind, heading back and just touching on the CNS specific modality, that would be helpful, and then we'll carry on.
Jackie, somehow I can't navigate back, so I
don't worry.
I will finish the presentation and I can then navigate back to that slide.
Yeah, and I think the major point that I was picking up really.
The point is that basically the CNS pathways were negatively associated with MS severity, which means that they were overrepresented in people who progressed slower, which suggests that they are neuroprotective, right? And synaptogenesis and Narayan signaling has been shown to be neuroprotective pathways. Can you hear now? Yeah.
You bet. Thanks so much, Bibi.
So the inflammation that we identified associated with MS severity is really compartmentalized inflammation, because we had from the same samples flow cytometry data and the soluble biomarker data, we could show that really there was inverse relationship. So for example, T cells, absolute numbers of T cells and proportions of T cells decreased in this cohort of untreated people. Similarly, NK cells decreased, B cells decreased. whereas the soluble biomarkers that are released by these cells increase, right? Which suggests that they are basically cells secreting these soluble biomarkers that they are not detecting in CSF because they are sitting somewhere in CNS tissue or meninges. Now, these different processes were differentially expressed in different MS patients. And unfortunately, our current classification of MS into these three categories tell you really nothing about which of these pathways that we link to MS severity are activated in the CNS tissue of which individuals. Finally, we validated models that predict all of these 9 outcomes that you see down here in the independent validation cohort. And of course, the effect sizes are always higher in the training cohort, a little bit lower in the cross-validation of the training cohort, but then substantially lower in the independent validation cohort. And the training cohort data do not predict what is the effect size in the validation cohort. And really the effect size in the validation cohort depends on the quality of the outcome. So that, for example, combi-wise, which we know is our best outcome, validated with really high effect sizes. And as I showed you, because combi-wise correlates so nicely with EDSS, we can simply recalibrate the data in the training cohort. So now we take the model to predict EDSS, and in the independent validation cohort, it predicts EDSS with the effect size that is highest compared to everything we were able to find in the published literature. In conclusion, this study confirms multiplicity and heterogeneity of candidate disease mechanisms in MS, and brings forward compartmentalized inflammation and its effector mechanisms as strongly associated with MS severity. And so I believe that we need to develop or keep on developing CNS penetrant anti-inflammatory drugs. Disease-induced re-expression of some of the CNS-specific processes seem to be neuroprotective, and perhaps we can pharmacologically strengthen these in people who do not re-express them naturally. We did not see any inflammation-independent neurodegeneration, and our results do support need for biomarker-guided patient-specific polypharmacy regimens. So thank you very much, and I am ready for your questions.
Terrific, Bibi. Thanks so much. So we've got a small group. We can just open up for questions by people just unmuting or popping into the chat if it works better for you.
I have a question, if it's okay. It was fantastic. I was really intrigued. So I understand correctly that you do see different patterns in the spinal cord dominant versus the brain dominant disease, right? So do you see, so you did that adjusted for sex and age also, because we often see, for example, that in males it's worse or it's stuff like this. So would you, based on what you've seen, would you suggest that some of the actual drugs would be better suited for the spinal cord disease versus the brain disease, or?
Totally. I don't think we have any drugs for spinal cord disease.
Yeah.
Right. So, you know, I think that basically this spinal cord disease was strongly associated with this fibrosis signature. You know, I think the question is, of course, chicken versus egg, right? So is it that some people have different genetic background that somehow, you know, makes them react to the same disease process by having predominantly spinal cord injury? Or is it that the lesions depend, you know, they just occur randomly, but when they happen in the spinal cord, the environment dictates how the inflammation, you know, how the inflammation evolves. So I think that you know, all of these are, of course, just correlations, and that's why we are running this TRAP-MS clinical trial, where we are assigning treatments based on the CSF biomarker profile, right? And then we are asking, are we inhibiting this biology that we are trying to inhibit? And if so, does the drug also then inhibit disability progression, right? So I think that that level of evidence must come from intervention. It cannot come from, you know, these studies. These studies are just hypothesis generating studies.
Mm-hmm.
Thanks. That would be so promising to be able to tailor for these like catastrophic cases where they have this spinal cord disease. Thanks. That's really brilliant.
Bibi, I have a question. How much do you think the identification of all of the fibrotic pathways is suggestive of really the potential of the spinal cord to repair compared to the brain? I mean, is that really?
Yeah, I mean, if you
know, and I come back to this idea of inflammation, bad, and we're saying inflammation is bad because everyone's out there targeting T cells and B cells and the lesion, and that's terrific. But the whole swing side of that is inflammation, repair, what we know needs to happen, whether it's fibrosis, which we don't want, or for some extent maybe we do. What do you think this means in terms of potential to repair and other things we need to address, whether it's a spinal cord lesion where we know there's some potential, or how to understand whether it's good or bad that it's happening?
Yeah, I mean, I think that there is beautiful literature from spinal cord trauma and animal models of spinal cord trauma that that suggests that, you know, astroglial component of the scar is actually not the worst one. That it's the fibrotic. It's when you have the mesenchymal component either through, you know, endothelial mesenchymal transformation, where you basically create fibroblasts and you're producing this, you know, dense extracellular matrix that really prevents repair, and that you have to target that process if you want repair to happen. And I think, you know, again, for me, the showing that these that you can't just from the kind of clinical phenotype determine what is happening in the tissue, you know, that's really important, right? And I like analogy with cardiovascular diseases, because nobody would just blindly give you red pill, right? If somebody gets stroke or heart attack, the first thing that the clinician is going to do is find which are the mechanisms that led to that stroke or heart attack in that particular patient. And if you find 3 of these mechanisms, you actually have to simultaneously address all 3 of them, right? And then you don't use one dose fits all and wait for the second heart attack to happen. You're adjusting treatment based on these biomarkers. And I think that in central nervous system diseases, we will have to get to the same precision, and I don't really see any other way to get to that precision other than studying CSF biomarkers.
Yeah. Alice?
May I ask a question?
Please do. I see also Cynthia coming up as well. So why don't we go Alice and then Cynthia?
Thank you. I'm Alice Laroni from Genova, Italy. Congratulations. I really enjoyed the
I
presentation.
can't hear you.
Oh, Alice, if we could get you to speak up a bit more, closer to the mic.
Yeah. Let me close the door.
That might
son
help.
is playing the piano, so maybe this is disturbing. Can you
You know, let me tell my dog that I'm fully protected from all neighborhood squirrels.
Can you hear me?
Alicia, we can hear you. It's a bit quiet. So just speak into the
Okay.
microphone and be as loud as you can.
Yeah, okay.
That's working. We'll just wait till Bibi's back from Squirrelville.
Yeah, and you know. I had to come. I told Jackie we have such problems with internet at NIH because NIH is being hacked. We get like thousands of hacks every day. And so our Zoom and WebEx is horrible. And that's why I teleworked so that, you know, I can have a good internet anyway.
Oh, well, Bibi, I take the squirrels and the dog barking to hear from you. So let's hear from Alicia.
Yeah, congratulations. I really enjoyed the presentation. It's really beautiful and interesting. A very basic question is, do you mean that progressive multiple sclerosis may be in the brain, the one with more brain disease, does share some mechanisms with the auto-inflammatory diseases more than with the autoimmune diseases?
I don't think our data are demonstrating that. You know, the data are showing that there are two components to neuroinflammation. One is adaptive immunity, and it was interesting that the B cell pathways and the upstream regulators related to B cells that I didn't have chance to talk about, they were only associated with MS severity, but not with MS progression. And indeed, you know, IgG index doesn't increase on the longitudinal CSF measurements, but the IL-17 pathways were clearly, you know, increasing, and they were also associated with MS severity. You know, we can't tell whether the IL-17 originates from CD4 cells, like Catherine was showing, or, you know, it can certainly also originate at least partially from innate lymphoid cells, ILC3s, right? So, I think there is quite a bit of literature indicating that IL-17 or Th17 immunity is very important in maintaining epithelial barriers. And I think the epithelial barriers are significantly affected in MS, and maybe, you know, that's one of the ways why IL-17 pathways are, you know, associated with MS severity. And then, of course, there is an innate immunity, right? So in the true autoinflammatory diseases, you don't see adaptive immunity, right? You see predominantly these innate immune mechanisms. I have very little doubt that myeloid lineage plays important role in destruction of CNS tissue in MS and many other CNS diseases. So I wasn't particularly surprised to, you know, see that signature.
Yeah. Okay.
Thank you
for the question, Alicia. And Cynthia has her hand up.
So thanks, Bibi. I appreciated a lot your talk. It was great, and I really like your studies. So maybe I can comment on this last part, because I mean, indeed, not all IL-17 mediated pathways are necessarily immune cell mediated. So we have shown in an annals paper huge upregulation of IL-17 receptor on astrocytes in chronic inactive lesions. There are no T cells around, very few myeloid cells, huge astrogliosis, and those astrocytes upregulate the IL-17 receptor and may react to IL-17 by release of nitric oxide. So this could set a link between the IL-17 pathway and the production of toxic molecules by the resident glia cells. So I really like the insights you can get from the CSF, but I have some issues with then application to patients. We cannot get longitudinal CSF samples from our—
Why not? Why not?
We can only have a CSF sampling during the diagnostic phase.
So Cynthia, I'm going to argue that there is absolutely nothing that prevents us doing that. What, the reason why we were not doing that is that we didn't have anything useful to monitor, right? We are taking healthy people, putting them to sleep to have colonoscopy. I do not believe that colon is more important for well-being of a human being than brain. I am a healthy volunteer. I'm one of those embedded healthy volunteers. I had 3 LPs over 10 years. I have LP Thursday morning and I have clinic Thursday afternoon. It's not a problem. It's not a problem. It's
no, it is if the ethical committees do not allow it.
No. It's a limitation that we are posing on ourselves, and I believe that it has everything to do with not having, everything. IRB looks at risk-benefit ratio, right? So if we show that the benefit of CSF outweighs its risk, then the IRB will approve the study, right?
Okay, so maybe there can be places where it is easier to do it rather than others, no? So, but in most of the cases, it is really hard to get sequential CSF samples. On the other hand, we had a study, I set the DOI in the chat, on PBNC transcriptomes from relapsing remitting primary and secondary progressive MS. And we applied their machine learning to those transcriptomes and demonstrated that indeed there is a very clear predictive signature of progression already present in the periphery. So I'm simply wondering whether we can think maybe of planning a study where we can also use blood transcriptome, which would be easier not to get several times a long time, together with the CSF.
Yeah. Yes, Cynthia, and we are definitely open to independent validation. To tell you the truth, I have been studying immunology as a fellow for many years. I could never validate things in the blood. And so I don't know, I haven't really, I have to admit I haven't seen your study. So it has been independently validated and you see the— Absolutely,
yes, on independent cohorts.
Yeah, so maybe there is. We would. I'm totally open to that possibility and totally willing to, give us give you blinded sample for validation or, yeah, certainly, if now I just sorry to say, even with my math background, right? This thing is relatively most sterile organ in human body, right? Th17 cells or immune signature that you find in blood vary so much with there was a beautiful 3-year study of a single PI looking at the transcriptomic signature, actually omic signature of blood. And what was really phenomenal was the amount of variation in these things, right? We get, we get subclinical infections, we get cuts, we get, I just can't see how that is going to be.
So give a look at the paper.
I will. I'm totally open.
Yeah. No, Cynthia, I think you raised a great point. I think we had Sarah Martin, but I don't know if Sarah stepped off. She had another question or had a question. Sarah, are you still here?
Oh, I am. Sorry, can you hear me okay?
Yeah, that's perfect.
Yeah. Oh, sorry, I sort of had second thoughts about asking it, but thank you very much, Bibi. That was a very inspirational talk. I'm just a postdoc trainee, so I was just wondering, given the breadth of pathways that you've identified and given sort of the move towards machine learning and big data, do you think there is still a role for measuring 1 or 2 protein biomarkers at a time and correlating that with clinical outcomes? Or do you think that that's a very sort of low yield research method at the moment?
Very good question. So once you have CSF, right? Once you have access to CSF, or once you draw blood, the technologies are now that you can measure multiple biomarkers. Yeah. I would always measure multiple biomarkers, because a single biomarker, you can just have a measurement error, right? If I have 5 biomarkers that participate in the same pathway and all are changing in correct direction, that is for me much stronger evidence, than having single biomarkers. So I'm not excluding a possibility that there is going to be a value of a single biomarker, but if you especially because of the amount of heterogeneity that we see among patients, I feel like you have to really measure multiple biomarkers. And not necessarily 5,000, right? In fact, many of these pathways are represented by a handful of biomarkers. But the models, we tested different modeling strategies, and we saw exactly what people are seeing on the levels of genes, which is that the proteins are affecting phenotype linearly, and that the effect sizes are really small, right? So these validated models that I showed you, we have maybe 600 biomarkers, and each one is not crucial. You can get rid of one and you lose very little of the power, but you get rid of half and you really lose the effect size of the model. Mm-hmm. So I think we are on the proteins, yes, the effect sizes are larger than genes, but we are seeing exactly the same that people saw on genes.
Okay, thank you.
I don't see anything else in the chat. And so what I did want to say is, I think, Cynthia raises some good points. I'm really thankful still that there's researchers out there doing the studies that they can. And whether it's because the NIH is able to do this in a manner that others cannot. And I certainly, as a foundational researcher, can appreciate that I'm not going to get access to CSF in a regular or meaningful manner through studies we have going here. And I think, if anything, this highlights the importance of collaboration, which is why I'm now working with Bibi to try to see what we can do from our foundational studies of identifying a marker that we associate with inflammation and see in, secondary progressive but not primary progressive MS, and to give that knowledge to her and say, hey, Bibi, can you check this through your data and see if it looks like it comes out or not? And then we decide what we can do further. And I think, opportunities such as this maybe enable us to think about things beyond our own labs and our own groups. And that's a big part of what we're trying to do here. What I really liked, and I'd like to highlight from today's session, was that foci in kind of 2 different areas related to inflammation in MS, where we're trying to dissect positive versus negative outcomes from inflammation, have us looking at molecules like IL-17 in a manner that may suggest it isn't so unfavorable. And so when we think about homeostasis and repair and inflammation and all of these things, we realize there's another aspect, and somebody out there is looking at it, even if we're not. I think it's wonderful. So with that, I think what we'll do is wrap up in the interest of time. And I'd like to ask everyone here today to join me in thanking our 2 speakers. On behalf of Maria, Hedrick, and myself, thank you for your time, and importantly, the recognition of the important work being done by our presenters today, and how they're really uniting some common themes in MS. So this is the first session of many to come, and we ask you to feel free to reach out to us with suggestions for topics or speakers which you believe may be fitting for this group. We're hoping to meet 4 times a year, and I think, you know, attendance can wax and wane, but with planning in advance, I know we can always get together a good group. So we look forward to hearing from you, and again, wish you all well in your day. Thank you again, Bibi and Catherine.